to Chapter 14. This is a big chapter, but it's really interesting. It's very diverse. We're going to be covering, we're going to talk about normal flora. We're going to talk about terms and features of infectious diseases and how a infectious disease progresses. And we are going to get into tricks and gimmicks that microorganisms have, as well as introduction to epidemiology and public health. So, lots of topics to cover. So, the first thing is going to be normal flora, and in order to understand that, you need to understand symbiotic relationships among organisms. So, you can have a symbiotic relationship between coral and algae, like in the ocean. You can have symbiotic relationships between people and their normal flora. You can have symbiotic relationships between algae and fungi. That's called lichen. So you can have symbiotic relationships between microorganisms and plants and animals, or among a couple of plants, or even among microorganisms, where one microorganism is in a symbiotic relationship with another one. So there are three major types. There's actually more than this, but here are the three major types: mutualism, commensalism, parasitism. So parasitism is rather self-explanatory. In this relationship, one is harmed. One of the organisms is harmed. The other benefits. In mutualism, they both benefit. And in commensalism, one is benefited and the other one is neither harmed nor benefited. And the reason I have an asterisk there is most of our normal flora and our relationships with them are commensalistic. So there's some examples here of different symbiotic relationships. Up at the top is a view of plant roots there. And notice the nodules. And inside of a nodule that has been enlarged, there are bacteria, a rhizobium, for example. And it converts N2 gas from the atmosphere in the soil that leaches into the soil, converts it into a usable form of nitrogen in the form of ammonia, which benefits the plant and the plant roots and its growth. Over here on the right, you've got relationships between microorganisms and termites. The little protozoan that you see right here lives inside of the gut of the termites, and it actually breaks down cellulose. It's got an enzyme called cellulase, which the termite does not have. So the protozoan actually enables the termite to eat wood and paper and other things that termites eat. Without this little gut protozoan, termites would not be able to get any nutrients at all from cellulose. They wouldn't be able to break it down. Its little protozoan benefits because it's got food that comes to it via the termite eating it. Down here at the bottom, you've got a lichen, which is a relationship between a fungus and an algae. And over here is a relationship between a tree and fungi. So down here you see some little white things sprouting out of the ground. These are called fairy rings. They're actually little mushrooms. They're kind of small. It's a small picture. The mushroom, hyphae, mushrooms are just moles, variations of moles, intertwine amongst the roots of the tree. And so the little ring of mushrooms develops at the outer edges of the roots of the tree, which are about equivalent to the leaves that are on the extremity of the tree. Obviously, we're going to talk about normal flora here. And that's the main thing that we're concerning ourselves with in symbiotic relationships. Okay, so here's some other examples of bacterial and animal symbioses. And the picture you're looking at over here is a cow, obviously, and notice there's a hole that's been made in the side of its body. It's called a fistula, and you could actually stick something into that and you would be right in the stomach. This is its stomach, the picture in the very middle. It's got a 4-chambered stomach, and one of the parts of its stomach is called a rumen. Now, of course, cattle eat grass. They chew their cud. That grass is cellulose. Cows don't have cellulase to break cellulose down. They depend on their gastric microorganisms that are mainly bacteria to break cellulose down. And they all live in the rumen. So the rumen is kind of like a big fermentation vat where the bacteria break down the cellulose. So, normal flora. What do they do for us? And where do you find them? And when do you get them? Well, requirements: moisture and food. And where would you find those? In your mouth and in your large intestine. So that's where the biggest populations of microorganisms are. If you look at numbers of microorganisms in plaque in your mouth or in saliva, or you look at numbers of microorganisms in feces, you'll find outrageously high numbers of cells, way over billions per gram of fecal matter. You have residents, you have transients. Transients come and go. For example, you give somebody money at the store, they give you change, coins, and the coins have microorganisms passed to you from their hands. When you wash your hands, transients come off. Residents, though, are the guys that always live with you. They're entrenched in niches in your body. What do they do for you? Well, or what harm as well as benefits? We're going to get more into that. But look at the metabolic contributions of your intestinal flora on the table. They break a lot of foods down. They make a lot of vitamins that you absorb and that benefit you. They produce a lot of chemicals that are beneficial. They also produce a lot of odors and byproducts that may make you gaseous or nauseous or things like that. When do you get them? Well, when you go toddling through the birth canal on your way to being born, you pick up vaginal flora from mom. The fetus in utero is sterile, sitting inside of an amniotic sac, unless the mother has an infection that's been transmitted to the fetus. We're assuming not. By the time the newborn is even less than a day old, it already has normal flora. Now it has very small amounts, but it's got the beginnings of its own normal flora population. And of course it's added to. Overall benefits of your normal flora, as I've already pointed out on the table, they contribute to digestive processes. They keep your immune system at a kind of a low simmer to activate it. They take up space all over your body that protects you against pathogens coming in and being able to set up house. Those are some of the main benefits right there. And why is it important to know what kind of normal flora you have around the body? I've got 3 reasons here. One is it suggests types of infection to expect after tissue damage. For example, if you're in abdominal surgery and inadvertently the doctor nicks your large intestine, he doesn't know it or she doesn't know it, you obviously don't know it, and you develop an infection during your recovery period, it would be obvious that the organism causing that infection is going to be one of your gut bacteria, probably Bacteroides, maybe E. coli, maybe a Clostridium. Those are major organisms that are in the gut. So there's some predictive ability then by knowing what your normal flora are in different areas of the body as to what types of infections you've got. It also is going to potentially determine a future type of infection. Okay, so look at the three groups of organisms down towards the bottom in blue. Organisms that are pathogens. These are organisms that are professional pathogens. They're always pathogenic, no matter in what kind of person. They have gimmicks. They have tricks to get around host defenses. On the flip side, you got the non-pathogens, and these are guys that almost never, ever, ever are pathogens. They don't make capsules. They don't make toxins. They don't form spores. They have no tricks up their sleeves. They have no gimmicks. They can't get around your immune system or your non-resistant factors in the body, like white blood cells. So they are wiped out relatively easy. Now, there are times when non-pathogens can become pathogens if the host's resistance and immune system are so low that they can't fight any type of organism. Then a non-pathogen can become a pathogen, and it's called an opportunistic organism. These organisms of your body can cause infections under lots of different circumstances. Sometimes it's just as simple as a hormonal change. So, for example, in kids that are going through puberty, 12, 11 years of age, and into the teens, there's an increase in oil secretion from the oil glands, and oil and lipids are favorite nutrients of staph. And you start to get a lot of staph infections, and staph infections are associated with pimples and blackheads and boils and things like that, and acne. So here's some conditions that can predispose you to normal flora infections. Okay, so we're going to take a little tour through different areas of the body and look at normal flora. You've got a kind of a list here. And if you look at the organisms that are on the list here, notice that there are a lot of recurrent guys. Staph and strep. Here's staph again. And strep. Strep. Some people also have staph in the GI tract, although it's not listed here. You also find Corynebacterium. Here it is up here on the skin. You find Escherichia coming up in different places, particularly down in here. So there are a lot of microorganisms in your body that are in a whole lot of different areas around the body. So where do you find the greatest numbers? Actually, I've already told you that. In your mouth, in feces. Vagina's got a very heavy load of microorganisms. You might think your skin is heavily loaded, but it's not. It's pretty dry on the skin, even though you sweat some. It's pretty dry, and it's also salty because of sweat. And so even though you have maybe 1 million bacteria per square inch of skin, that's pretty low compared to 10 to the 14th cells per gram of feces. Areas that are sterile, areas of the body that are closed off to the outside. So blood, spinal fluid, synovial fluid, pericardial fluid, pleural fluids, all of those fluids in closed cavities. Muscle should be sterile. Heart muscle, smooth muscle, skeletal muscle. The nervous system should be sterile. So, let's go through some of the parts of the body. Skin. Here are some of the organisms that you find in the skin listed. Again, you see staph, and you see Corynebacterium, which is, let me spell it out here. This reference right here to diphtheroids, that's Corynebacterium. They're commonly called diphtheroids. In fact, this is Corynebacterium right there. It's a short gram-positive rod, kind of odd shapes, sometimes called drumstick shapes. Here's your typical staph, and here's a fungus that's often on skin. So if you go over to the right to upper and lower respiratory tract, here is staph again, a nice scanning EM showing staph attached to cilia on the top of pseudostratified ciliated columnar epithelium that you find in the respiratory tract. Here is Neisseria. For example, Neisseria meningitidis we've already talked about in class. There's some other relatives of Neisseria as well as other species of Neisseria that are found in the mouth that are non-pathogenic. Here's a typical strep there. By the way, you might wonder what the difference is between upper and lower respiratory tract. The boundary line is your larynx, your voice box, so upper is above. And of course, the higher up, the greater the number of flora. And as you get down into the lower respiratory tract and you move down the bronchi and into the bronchioles and down to the lungs and the alveoli, you're almost, not quite, but almost to an area of sterile habitat in the alveoli. You shouldn't have very many organisms down in the lungs. Not quite sterile. So moving over in the digestive system, this is where it gets kind of messy because there's so many different areas of the digestive tract. The habitat in the mouth is a very different habitat than the area of the stomach, which is highly acidic. And if you think about your mouth, you've got a lot of different habitats, even within the mouth. You've got the open areas of the mouth, you've got the areas inside the cheek, you've got the areas under the tongue, on the tongue, you've got on the teeth, you've got the crevices between the teeth and the gum, and those areas are almost anaerobic deep in those crevices. So lots of different habitats, even within that one large habitat. You go down into the stomach, it's highly acidic. We move over into the small intestine, it starts to become more alkaline and a little bit more conducive to bacterial growth. And then we get down in the large intestine, and life is really good. A lot of undigested nutrients. There aren't very many enzymes that are down in the large intestine. Remember, if you've had A&P, you know there are a lot of digestive enzymes in the stomach and in the small intestine, and those digestive enzymes can harm bacteria. But in the large intestine, everything that's going to be digested is digested, except maybe for some water reabsorption and a few vitamins. So there's nothing to really kill bacteria. So if you look at the organisms going down the digestive tract, to the left of the stomach, you see lactobacillus. You see a few organisms in the small intestine, which is the pink tan area. And then the large intestine in purple, look at all the organisms listed. And this is a short list. It's not even the whole list. Another difference among all the areas of the digestive system is how much oxygen is available in your mouth. There's a lot. But you get down into the small intestine, and in the large intestine, it's pretty anaerobic. So you're not likely to find any strict aerobes down there. It's just not the right environment. Some of the organisms you see over here, here's strep again. Here's E. coli and all of its relatives, its cousins. And here's Clostridium, which is a gram-positive rod. Okay, we move down into the genitourinary tract. I've combined them together here. Because, for example, in men, the urethra is both a conduit for semen as well as a conduit for urine. So it merges together in men, although it's separate in women. So the organisms you see in this area, here are three common ones. Here's Corynebacterium again. Here's Staphylococcus. And here's Candida yeast. And you'll notice, looking at the area of the urethra, here's the urethra, here's the vagina, that again there are a lot of bacteria here, many of which are found in both areas. Here's just a look at where you find the microorganisms and the recurrent pattern where you find them. So look up at the top, you'll notice staph is up there, and you'll find staph all across the board, from skin to the front of the eye, the nose, the pharynx, the mouth, the lower intestine, urethra, vagina. Other ones that you find in areas, look in the middle, you see the coliforms like E. coli. It's pretty much found across the board, not necessarily everywhere, but most places that you see listed here. And then its relatives like Proteus and are found in lots of areas. Then you go a little bit further down, about fourth down from the bottom, you see Corynebacterium again, all across the board. If you look at the streps, like 6, 7 down, you see a lot of species here. Different species in different areas, but generally if you added them all up, you'd find them all across the board in different areas. So some terms that you should know about. So we're leaving normal flora, and we're moving more towards features of diseases now. So we've already talked about colonization in class. Let's start with colonization. When the organism gets into your body and it starts to reproduce in an area, you are said to have been colonized. The organism has set up shop in or on your body. When it starts growing and it's now entrenched in your body, you are now infected. In fact, we are infected from birth to death. You spend your entire life being infected. Now, you tend to think of infection as having host injury, but really, that's the term disease, because we're all infected right now. Infection just implies growth of a microorganism. Disease is infection with host injury. So you've got a couple of terms here called portal of entry, portal of exit. How does the organism get into the body? How does it get out? Sometimes it's the same and sometimes it's different. Let's go flu. You can inhale influenza virus. You get influenza or flu, and you cough it out or sneeze, and you exhale it through the respiratory tract. Same way in and out. But of course you can also pick up flu by handling objects, so direct contact, but respiratory portal of exit. Let's take HIV. You can get it by sexual intercourse, and you can then give it by sexual intercourse. Or you can get it by sexual intercourse and you can give it by blood. Or you can get it by blood and then you can transmit it to a fetus that you're carrying. Signs versus symptoms. Symptoms are subjective. So you may think you have a fever, but unless you have a thermometer to record your body temperature, it's subjective. You may not really have a fever. You may just feel like you're hot. So that's a symptom. The doctor wants factual information about your body temperature. We'll use a thermometer. That's a sign. So a sign is objective, and a symptom is subjective. Has to do with what you feel like, what you are complaining about, or what others see in you. For example, somebody might think you're nauseous because you're pale, or you complain of having a queasy stomach. Asymptomatic and subclinical, same terms. We'll get back to that. And etiology is the origin of disease. Koch's postulates. We went over that in Chapter 1. Wanted to give you some examples of problems with Koch's postulates. Not everything fits those postulates. So, reminder, in a group of organisms that have similar symptomology and signs of a disease, there will be one cause. 2, you can sample some of the organisms, some of the animals or plants, whatever kind of disease we're talking about, take the samples, grow the organism in a laboratory using media, running tests, microscopy, blah, 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 isolate it. 3, you take your isolated organism, you put it into healthy, susceptible animals, you recreate the disease symptoms seen in the original animals. 4, you re-isolate the specimen from those newly diseased animals, you run tests on it, verify that it's the same organism identified in step 2. So let me give you some examples where it doesn't work, or that Koch's postulates don't necessarily work well. Remember that the first postulate says that when you have a similar symptomology in a group of animals, there must be one cause. Well, that's not always true. There are multiple causes of pneumonia. So we could have 10 people in a hospital having pneumonia and having similar symptomology, because the symptoms of pneumonia are rather similar: coughing up blood and sputum, and problems with breathing, and pulmonary edema. Sounds like you're kind of breathing through water when you put a stethoscope up to their chest, things like that. But you could have a fungus causing the pneumonia in one person. You could have a couple of other people having strep pneumonia, which is bacteria. You could have another one having viral pneumonia. So in actuality, you could have multiple organisms all producing similar symptoms. Another example, the second postulate says that you can take a specimen and grow the organism in a laboratory. Well, you can't always do that. Two examples. Mycobacterium leprae, which causes leprosy, you have to grow it in a laboratory. There is no medium that has ever been developed that you can grow that bacterium on. Another example, the syphilis bacterium, Treponema pallidum, never been grown on a medium. They have to grow the organism in rabbits. Usually rabbits are a choice of the organism. Usually there's a test animal that you can use to grow it in, but it doesn't quite fit Koch's postulate of number 2. Okay, pathogenicity versus virulence. You know what a pathogen is. You know what pathogenicity is. Virulence is the degree of pathogenicity. So you have organisms that are more pathogenic and organisms that are less pathogenic, but they're both pathogenic. They both cause disease. So the term used is usually virulence. Those pathogens that are mildly pathogenic are not very virulent. Those organisms that are very pathogenic, very virulent. So we're going to look at 3 different ways, tricks, that organisms have to produce pathogenicity. Toxins, being able to avoid host defenses, that's called invasiveness. Those are the 2 major ways, and also some features of the organisms, like ability to form spores. So here's a look at some virulence factors. These are the gimmicks and tricks that microorganisms have to get around your host defenses. Adhesion factors. Some organisms make proteins that allow them to stick to your cells, or they make fimbriae that allow them to stick. So there are a lot of different adhesion factors for stickiness and attachment, which allow the organism to immobilize and set up a focus in a particular area. Enzymes. There are a lot of enzymes that a microorganism can produce that are excreted out. Collagenase, which allows the organism to spread through collagen. Now remember, collagen is protein of the body. It's in connective tissue. If you've had A&P, you've heard of collagen a lot. Collagenase allows an organism to spread around tissue. Coagulase allows an organism to coagulate or clot blood. Homolysins allow an organism to hemolyze blood. So here's a look at hemolysis. You're looking at a blood agar plate. That plate is streaked with staphylococci. You can see the white colonies. The plate is sitting on newspaper. And so you can see these kind of clear areas around the colony, kind of clear yellowish areas. And if you can see it really well here, in fact, it's broken up all the red blood cells, allowing the plate to almost become invisible, so you can actually see the print of the newspaper below. What you're looking at up here are 2 tubes of plasma that's been removed from whole blood. This is coagulase enzyme that's clotting that plasma. And then a lot of organisms make toxins. We're going to talk about this in more detail, exo and endotoxins, that offensively destroy the host cells and tissues. I didn't mention yet antiphagocytic factors. Take a look at the capsule stain right here. You've seen one in lab. Also, some microorganisms, even though they may be phagocytosed and they're sitting inside of a white blood cell, they can make chemicals that kill the white blood cell, thereby freeing them. They're called leukocidins. Cide for cidal, kill, leuko for leukocyte, kills white blood cells. Not going to get into any of this, but wanted to mention inside of that yellow box at the bottom, a lot of these virulence factors that I mentioned are carried on plasmids. Remember, plasmids can be transferred horizontally to other cells as well as vertically to daughter cells. So it's like inheriting a Bible from your family, kind of. Maybe a bad analogy, but anyway. Okay, here's a look at different kinds of chemicals that I just kind of went through. Here are a short list. It's much longer than this. These are some of the enzymes and chemicals that are gimmicks of microorganisms that help them get around in your body and do destructive things. Look at the organism examples that can make them, and you'll see recurrently Staph aureus, Strep pyogenes, and Clostridium. Clostridium, there are a lot of species. Clostridium perfringens, which causes gas gangrene. Clostridium tetani causes tetanus. Clostridium botulinum causes botulism. Staph aureus causes toxic shock syndrome, impetigo, boils, abscesses, any number of things, wound infections. Strep pyogenes, or what are called group A beta hemolytic strep, cause strep throat. Also impetigo, erysipelas, kidney infections, rheumatic heart fever, thinking what else? Sometimes called flesh-eating bacteria, also can cause toxic shock syndrome. So these are big professional pathogens you see on the list. And I'll talk about the picture that you see at the bottom in lecture. I don't want to get into that. So I'm going to stop here because I don't want to get into toxins yet.